Hello, this is Madame Biology, and we are going to be going over the 4 biological macromolecules today. All right, let me share my screen. Okay, so this is the slideshow that I've prepared for biological macromolecules. So the 4 biological macromolecules, or the building blocks as I like to call them, are carbohydrates, lipids, proteins, and nucleic acids. So let's start off with carbohydrates. They're essentially just sugars. They can be simple or complex. And what I mean by that is like simple is monosaccharides, so like one unit, and then complex are polysaccharides, disaccharides, and oligosaccharides, which are multiple units. Disaccharide, di is two, so that's two. Oligo is like small chain. I'm not sure what the exact root word means, but oligosaccharides and polysaccharides are pretty similar, so it's okay to just think of them as chains. So this is a glucose monosaccharide. And then this is the entire thing is maltose, which is a disaccharide. So how like these monosaccharides and polysaccharides kind of associate with each other? There's two different processes to that, dehydration synthesis and hydrolysis. So dehydration synthesis is the process of removing the oxygen atom and the hydroxyl group, which is OH, to make one water molecule and a disaccharide. Hydrolysis is the process of adding an oxygen atom to to a monosaccharide and the hydroxyl group to the other monosaccharide to split them apart. What I mean by that, I will demonstrate. So if this is two polysaccharides, we can do and then plus H2O. We can do hydrolysis to split them. So this will have an O or an oxygen, and then this will have an OH. So this is kind of what hydrolysis looks like. And then dehydration synthesis, what that would look like is, oops, let me start off with this. You'll start out, and then to put them, oh, I'm sorry, this is not it. Plus, you're going to combine these two to make a molecule of water and put these two molecules together. So these linkages, they're called glycosidic linkages, and glyco means sugar, so that's a good way to remember that. And carbohydrates pretty much have a 1 to 1, 1 to 2 to 1 ratio, and what that means is that they'll have a ratio of 1 to 2 to 1 with carbon, hydrogen, and oxygen respectively. So if we take C6H12O6, which is glucose, this would be 1, 1, and then 2, because 6 times 2 is equal to 12. So this would be 1 times 6, 6 times 2, and then 6. Okay, and then isomers. This is kind of like another sub part of carbohydrates. It's basically the same chemical formula, but a different structure. There's from the ones that I know the most about, there's three different kinds. There are structural isomers, which they're the same chemical formula, it's just the way they're arranged are different. Cis-trans isomers, it's just the arrangement around a double bond is different. So as you can see, this is X. This is like this molecule, hydrogen, hydrogen, and then the single bonds can rotate on this axis, but the double bonds can't. So that's what kind of gives them that structure. And then, forgive me if I say this wrong, but enantiomers, I I don't think I said that right, but that's just the 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 way that it's oriented, left or right, around an asymmetric carbon. Okay, so now quickly onto the ring structure of sugars. So hex stands for 6, is a sugar with a 6-carbon ring, and then pent is 5, that prefix. So then glucose would be a 6-carbon sugar because this would be 1, yes, 1, 2, 3, 4, 5, 6, then 1, 2, 3, 4, 5 for fructose. All right, so now on to proteins. So proteins are made of made up of building blocks called amino acids, and amino acids are coded in, or they're coded for by mRNA. And amino acids are connected by peptide bonds to make a polypeptide, and these proteins are coded for by DNA. So DNA to mRNA to protein, this is the central dogma of biology, this process. And going from DNA to mRNA is transcription. Going from mRNA to protein is translation. And there can be many different kinds of proteins. They're a really versatile group of macromolecules. And so they can be enzymes, hormones, defensive proteins. They can be a lot. So they proteins are very, very important in biology. One of the most important enzymes in all of biology really is Rubisco, which will be covered later on in the AP biology curriculum and overall, but it's a very important enzyme. So proteins are very, very important. Defensive proteins would be proteins like antibodies, which are a part of really important part of your immune system, and hormones like insulin. Insulin is classified as a protein, I'm pretty sure. So yeah, proteins are just very, very important. So now on to the building blocks of proteins. This is the structure of amino acids. So an amino acid will always have this central carbon and then this hydrogen attached to it. And then it will have this carboxyl group and then this amino group. It's called a carboxyl group because this group is a carbonyl group. Put that out. Carbonyl. And then this group is a hydroxyl group. So when you kind of smash those two together, you get a carboxyl group. And then this is an amino group. This R group is the most important part. This R group is different for every amino acid, and that is what gives amino acids their unique properties. So these R groups, based on the arrangement of atoms, they can be hydrophobic, hydrophilic, polar, non-polar, charged. They're very diverse, and that is what makes proteins so unique. So the structure of proteins, there's four different kind of levels. So the primary structure is just the chain of amino acids. Secondary structure is the hydrogen bonding between amino acids to make the two most common structures, which are alpha helices and beta pleated sheets. Yes, I go into that in the next slide. And then tertiary structure is deal mostly in disulfide bridges and more hydrogen bonding to put peptide chains into polypeptides. And then quaternary structure is multiple polypeptide chains coming together to make a protein. And this doesn't happen in all proteins. This only happens in some, and it's not fully understood yet, but it is thought that proteins called chaperonins might have a help in quaternary structure. Yes, okay. So this is kind of what a chaperonin looks like. An unfolded polypeptide goes into the cylinder. This kind of cap closes on it, and then this kind of creates an environment for the protein to fold in the ideal way, and then the cap pops off, and then the protein pops out. So this is what an alpha helix and what a beta pleated sheet would look like. Oh yeah, as you can see, hydrogen bonds, hydrogen bonds, hydrogen bonds, and hydrogen bonds. Okay, so on to a also an important part of proteins, which is denaturing. So proteins can change their hydrogen bonding based on pH and temperature. So this can create a dysfunctional protein, but in some cases it can also just not have an effect on the protein. And proteins can become permanently denatured, but they can also become temporarily denatured. And if the pH and the temperature goes back to the ideal conditions, then then like then the protein will come back into its normal form. Okay, so now onto the next group, which is lipids. So lipids are fatty acids. An example of this would be triacylglycerol, which is this picture right here. And there are ester linkages between lipids. So this would be an ester linkage. And van der Waals forces, which are weak, non-ionic forces, hold these lipids together. That's more of advanced chemistry. So then these ester linkages, which is what's right here, is what kind of uh keep this part and then this part together. This is the fatty acid, and this is the glycerol. So the glycerol and then the fatty acid are held together using these ester linkages. Okay, and then yeah. So there's one glycerol for three fatty acids, and that's what gives its name triacylglycerol. Okay, now an important subtopic of lipids are phospholipids. So phospholipids are an important part of the phospholipid bilayer, which are in cell membranes, and they make up most membranes. They have a phosphate, and then they have a glycerol tail. Oh, they have a glycerol right here. They have fatty acid tails, hydrophilic head because the phosphate is charged, and then hydrophobic tail because the fatty acid is not charged. So what makes this membrane so ideal is that this prevents the membrane from just dissolving in these charged conditions outside and inside of the cell. But then the hydrophilic heads make it so that these molecules can interact with the membrane, but the membrane also just doesn't melt away from it just kind of falling apart. Okay, unsaturated versus saturated fats. So saturated fats are solid at room temperature, and there is no double bond here, so there isn't this bend. And then unsaturated fats are liquid at room temperature, and there's a double bond here. So, and there's one more carbon that's creating this double bond, and that's what creates that bend. And as a very, very general thumb rule, saturated fats are mostly animal fats, like butter. Can't think of anything else right now, but yeah, butter. And then unsaturated would be oils, so like olive oil, sesame oil, canola oil. Yeah. Okay, so now on to the next group, which is nucleic acids. So nucleic acids create proteins with the help of mRNA, ribosomes, and tRNA. So nucleic acids, that group has DNA and different types of RNA. So rRNA, tRNA, mRNA, siRNA. There's also pRNA, that's like a different group. And then there's also different types. I wouldn't say different types, but more like different locations in which they're found, such as mtDNA, which is found in the mitochondria, and cpDNA, which is found in chloroplasts. And that's endosymbiotic theory, which hopefully will be another video on this channel. And this is kind of the process of going from DNA to mRNA to protein. So mRNA is made in the nucleus in eukaryotes. In prokaryotes, this entire process happens kind of back to back because prokaryotes don't have a nucleus, so this entire process kind of happens just back to back, and there's no like nucleus to separate it. And the mRNA will leave via this nuclear pore right here. The ribosome will synthesize the protein using mRNA and using tRNA, which will bring the amino acids. So the bases of DNA are adenine, thymine, cytosine, and guanine. A pairs with T and C pairs with G. So adenine pairs with thymine, cytosine pairs with guanine. The bases of RNA are adenine, uracil, cytosine, and guanine. Adenine pairs with uracil, cytosine pairs with guanine. This is the base pairing. And that's what the base looks like. So this is the nitrogenous base, which is adenine, thymine, those bases, and then a sugar, and then a phosphate. Okay, so this is a quick vocab about nucleic acids. So a gene is a section of DNA that codes for a protein. Alleles are variations of the same piece of DNA, one from the biological mother and one from the biological father. Chromosomes are a long DNA molecule that is wrapped around organizational proteins called histones, and genome refers to all of the DNA in the cell. Other than other than mtDNA and cpDNA, I'm pretty sure. So genome is just the genes in your nucleus. So I have this small segment on basic Mendelian genetics. So let's start off with Punnett squares, which are kind of the very building block of genetics. So this uppercase A stands for red hair. This is just an example. And then this lowercase a stands for black hair. So these big A and small a stand for different alleles. So the big A would be the dominant allele, and then the small a would be the recessive allele. So some rules to basic Mendelian genetics. Recessive alleles can only be expressed when paired with another recessive allele, and dominant alleles can only be expressed with another dominant allele or with the recessive allele. And homozygous is when two same alleles are together, and then heterozygous are when two different alleles together. There are many more different types of Punnett squares, like dihybrid crosses, trihybrid crosses. This is just the most basic version. So, a quick example about homozygous and heterozygous. This this quadrant here would be homozygous, and then this quadrant here would be heterozygous. Okay. As I mentioned before, another type of a Punnett square is a dihybrid cross. So a dihybrid cross is a way to test if the parents are heterozygous or homozygous, and two traits are tested. So the steps are to identify the genotypes of the parents, use the FOIL method to determine all possible combinations, organize them into a Punnett square, drop down the combinations and combine, which I'll explain in the example in the next slide, and then answer the question if there's like a specific thing that the question is looking for. So this is a sample problem. In dogs being clumsy, this is taken from Khan Academy. In dogs, being clumsy, capital C, is dominant to being cool, and being dazzling is dominant to being docile. A female dog that is heterozygous for both the C and D trait is mated with a male dog that is also heterozygous for both traits. Both the C and D traits sort independently from each other. That is a separate piece of information that we don't really need to worry about right here. So the genotype of the mom would be CCDD and the dad would be CCDD, the same thing. And then this is the FOIL method. So if we do CCDD, C to D, C to D, C to D, C to D, that's it. That's just the FOIL method. You need one allele from each trait, that's it. So this is basically what the FOIL method is. So then you end up with CD, 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 and then CD. And the next steps are on the next slide. Okay, so this is how I set up my Punnett square. Usually this like order is the format that is most used because it's the one that makes the most coherent sense. Like you start off with a two dominant, you go one dominant, one recessive, one dominant, one recessive, and then both recessive. But really, any combination will work as long as it's consistent on this column and then on this column. I mean this row. Okay, so what I mean by drop down is you just take this allele and then this allele and then you combine them. So these two dominant alleles will come together and then these two dominant alleles will come together. That's what dropping down means. Okay, did I explain? Oh no, this is practice problems. Yes, so then I will explain a very interesting ratio that you will see in dihybrid test crosses. Oh, I forgot to explain something. Oh, okay. So the name dihybrid, which took me a very long time to realize, was the name dihybrid comes from the fact that di is two, so you're testing two traits, and then hybrid means that you're testing two heterozygous parents. So yeah, just that's a good way to remember what a dihybrid test cross will look like. Okay, so now on to phenotypes. So the phenotype of a dihybrid test cross will always be 9 to 3 to 3 to 1. And I'll explain this. So this 9 comes from the fact that you will see all of these 16 possible combinations. 9 possible combinations will have both traits being dominant. As you can see, CCDD, 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 CCDD. So these are all dominant for both traits. And then this 3 is the fact that one trait will be dominant for this one, This one trait will be dominant and then one trait will be recessive. And then this 3 is that the other trait will be recessive, and then the previously recessive trait will now be dominant. And then this 1 is that both traits will be recessive. And this will definitely be used in later questions, but yeah, that's a very cool ratio and very important ratio. All right, so I have some practice problems here, and I will link these in the description. All of these are from Khan Academy. Most of the information in the slides is from my own knowledge, and any other gaps have been filled by using Campbell's Biology, the 10th edition, online version from archive.org, because I'm not rich enough to buy the entire textbook. So that is the end of the video. Stop sharing. Thank you for watching, and I hope this helps. And yeah, thank you.